Hello. Thank you for inviting me. I just want to say I'm really sorry I can't be there. COVID prevented that. It's an amazing place you have there and an amazing community. And um, again, I'm really sorry I can't be there. I hope I've tried to put together a lecture for you that I hope will be entertaining and open up this session. So I titled this lecture Reverse Engineering Human Visual Intelligence, but I could have probably titled it something more like this: computable modeling as hypothesis building and testing for application relevant scientific progress. Now, the first title is more punchy, so I think we'll stick with that. But I hope it'll become clear through my talk why what I'm trying to say here in terms of both scientific philosophy that I'll talk about, but also give you some sense of where progress is and vision and maybe how we can go forward together as a community. So let me first start off with the most important slide of any talk I give, which is anything I'm going to tell you today that comes out of my lab is really not my work. It's really the work of these amazing women and men. And I will try to highlight some of them along the way. So our lab, and I think many of you in this session, are really interested in visual intelligence broadly construed. Now, I like to show this slide this image at the beginning to say, what are all the amazing things that we can do just looking at an image like this one, reminding you that the only thing striking your eyes right now is a pattern of light energy displayed across the screen. Yet we make inferences about what's out there in the world quite rapidly. Are there objects in the world, people, cars, trees, buildings, and other predictive inferences like where is it safe to walk? It's probably more safe to walk on the left than the right, for instance. And you do that effortlessly and quickly. You still do that far better than current machines, and we still don't fully understand the neural mechanisms that allow that to happen. And really, that's the goal of our work and others in this community, is to understand those mechanisms in an engineering terms. Now, we can't study that problem all at once. And what our lab has tried to do is to narrow this problem to a smaller problem. And we do that by first starting by thinking about that you don't digest that whole scene at once. In fact, you have high acuity at the center of your gaze. So if you were fixating at that red spot, you'd have high acuity around this object, this particular car. And the way that you sort of digest that scene is by making lots of eye movements around the scene to start to take in various glimpses of the scene and assemble them into those inferences that I was describing earlier. So this process involves an unfolding in time and to get to full visual intelligence. And I'm not going to tell you today about all of that. We started with something that was tractable, and that sort of paints the frontier of where the field needs to go next. But we're going to tell you the progress that we've made in the more limited domain as hopefully inspiration for the broader questions of intelligence and vision. So that limited domain we refer to as core recognition. That's just what you're doing in the central field of view, the first 200 milliseconds in which an image like these ones strikes your center of gaze, being able to identify and make inferences about what's out there, the latent content, for instance, the object identities, cars, people, and signs, other latent content like the position, pose of these objects. This is something we broadly refer to as a domain of visual intelligence called core recognition. And we think, again, it's core to visual intelligence, but not all of visual intelligence. But it turned out to be a remarkably good place to start to understand processing along one part of the visual system called the ventral visual stream that I'll tell you about in a minute. Okay, with that context, I want to just also remind you that we don't just measure those things neurally that I'll show you in a minute, but we need to ground this in kind of what are the kinds of things humans actually do, not just show you images, but test humans in doing this. And so we test many humans, we and others, in tasks like this one, being able to identify latent content that I just described. And here's an example task you can try yourself if you fixate the dot. Hopefully you noticed an image came up and you were able to choose that it was more likely a bird than, say, an elephant. And here we go again, same thing. That was more like a face on the left, and you would choose that rather than the car. And here's another example. One more. Again, that's a natural photo of a bird. Okay, so those are just examples to demo your amazing abilities. This is another organism that's closely related to us. This is the non-human primate, the rhesus monkey, who we make extensive use of in my lab and others to study visual processing. This animal is doing the same task that you were just doing, except triggering it with a touch screen, getting a test image at the center of gaze, and then choosing among a pair of objects. And the animal is able to do this over dozens of objects, doesn't need pre-queuing, just like you didn't need pre-queuing, yet is remarkably good. Those are the green flashes, getting those trials correct, but also not perfect. Those are the black squares. He got that one wrong. And you and I are also good, but not perfect. And one of the things we learned by studying that species, our close cousin, and perhaps not surprisingly, is that when you look at the patterns of confusion that that species makes and our species makes, us on the left, that monkey species on the right, you see that these are patterns of confusions over objects. And all I want you to take from this is that these patterns look remarkably similar. In fact, they're very hard to distinguish. They're almost quantitatively exactly matched between these two species. And again, that fits with an idea of conservation of mechanisms across evolution. And this is just to show you this at the object grain. We've also done that at the image grain that I'm not showing you here. But the big picture I want you to take from this is that this allows us to think about, on the right, primates as a group, say primates here, just Homo sapiens and rhesus monkeys, as an equivalent group with respect to these core visual recognition tasks, similar mechanisms, almost quantitatively exact behavioral performance. And on the left, computer vision systems, which we take as sort of models of engineered models of what you may want to someday be primates, but are not yet primates. And I'll give some examples of their failures along the way here. And this really motivates us to say we can use a model system of a non-human primate, but we can compare it with also leading computer vision systems, and also now engineered models that we take as models of the mechanisms. So again, getting to this theme of this symposium, the dialogue between the things on the left and the things on the right. Okay, so we study primates not just because we like monkeys, but because many people have worked before us to show that this is a very good model of the humans in many other ways in the visual processing stream. And we know a lot about the monkey visual stream. In particular, we know a lot about the cortical areas involved in central vision, the so-called ventral stream, shown here as colored areas on the monkey brain shown in the right. And this species affords us much greater access to the neural hardware than the species on the left. And so therefore we use it again as a model system. Now, this is the example of how we conceptually think of what's going on in the ventral stream when an animal such as you or I or that monkey is watching a sequence of images such as the one shown here. There are population patterns of neural activity being generated all along its ventral stream in each of these cortical areas, millions of neurons firing in each particular area. And it takes about 100 milliseconds for those patterns to propagate their way up to the top of the ventral stream in the so-called infratemporal cortex area, IT. But I want to emphasize this isn't just about IT. It's a network of areas processing this information in both a feed-forward and a recurrent fashion. Okay, we're especially interested in this area, this sort of network of areas, because it's thought to be critical, in fact, known to be critical to core recognition tasks. Lesions in this part of the brain produce deficits in the tasks that I just showed you and other sort of related tasks. I also want to highlight that this is not just neural, of course. You don't just do neural processing for no reason. You ultimately have to get to behavioral output. And this ventral stream projects to regions involved in decisions and actions that I won't talk about today, but it's well positioned to drive the kinds of behaviors that I just described. And in fact, we have a lot of evidence to suggest it does drive those behaviors. So just by way of background to introduce you to IT cortex, again, you can think of the top of the ventral stream. It's producing patterns of neural activity, again, schematically shown in red. What we do is we record from monkey neurons using, in our case, chronic recorded implanted electrode arrays. We can record hundreds of neural sites in any given animal, in this case from IT cortex, using these chronically implanted arrays implanted during a sterile surgery, in which we can then later record the same neural sites for many weeks or months, which turns out to be quite valuable in terms of constraining and building models. And one of the ways that we think about the data coming out of the neural response data is we take the neural firing rates as a starting hypothesis as the most useful way to look at the data. So we take firing rates over small windows of intervals of time, and here I'm showing you examples of mean firing rates over narrow windows in time from IT cortex in response to one image. And the green color, high green is high response and black is low response. And all I want you to see is there's a vector of responses across here, hundreds to thousands of neurons that we can assemble across many monkeys to say this is the sort of typical IT population vector that comes out for that particular image, in this case, and one image at the top shown here. Of course, we don't measure just one of those vectors. We measure those population vectors for many images. Here's 8 example images. And notice that the population patterns look different for each of those images. And as I mentioned, we record for many months. We can record thousands of images, and we can record many repetitions of each image to get very high signal to noise on the mean firing rates, even with narrow time windows. So we get a very good estimate of what we think is the important representational space in IT cortex and the other visual areas that I outlined earlier. But by way of background, one of the things that we know, and I won't have time to tell you about this work, is that we and others have shown that if you look at this particular way of looking at the data, the mean firing rates over narrow time windows, again, 10 to 100 milliseconds, that those population patterns are sufficient to explain the behavioral patterns that I showed you earlier, or that I demoed for you earlier. You just need linear decoders to be able to apply on top of the IT data to explain this. And those same things do not work when applied to earlier parts of the system. Okay, that's by way of background. We refer to this IT response patterns as an IT code, not because we understand it in all details, but because we have extensively tested hypotheses of its linkage to behavioral reports and tasks like core recognition and related latent variable inference tasks. Okay, so this finding, all I want to you should take from this part of the talk is this finding makes these neural patterns cognitively relevant. And we also have a handle on the mean fire rates as being the useful measure of the neural data. And both of those facts are important to moving forward, but you should just take that as background. So the real thing I want to talk to you about for the most of the talk today is about how this code is computed from the image. So there's all these intermediate representational spaces that are computed, and somehow the image is transformed up through this network to these useful patterns in IT cortex. And this problem we knew this was a long-standing problem that could not be solved by one lab. It's a long-standing systems level science problem. How are we going to figure out, we as a field, not just us as a lab, even in the ventral stream, even in a more limited behavioral domain like the one I said here, how is we as a field going to actually take on challenges such as this, these complex challenges? That's really the thing I hope we can all do together. And we're going to tell you how we've been trying to set the stage for communities to work on problems like this. So how do we proceed? Well, let me sort of give you a kind of a perspective that we have in our lab. And this is sort of the philosophy of science view that I want to convey. The perspective is this. So you have science on the left, which measures phenomenology, and you have engineering on the right. And the way you get started on sort of understanding a system like the one I just described is you have to start somewhere. You build a hypothesis, which for us is some starting ANN models. Now, what's an ANN? An ANN is an artificial neural network. I'm sure all of you have heard that or know that. I'm not saying that today's artificial neural networks are the only models you should build, but because we're neuroscientists, we need these models to map the neurons. So we're going to build with something that has some kind of artificial neural networks in it. So it may not be today's ANNs that turn out to be the best models, but we need to have ANNs in the modeling framework. So we have ANNs as starting models, and then we're going to evaluate them. Let's build some, let's try our best, let's evaluate them against the neural data and the behavioral data and see how well they work. And of course, no models are going to be perfect, so we're going to come back and we're going to use those evaluations to hopefully build some better ANN models. And there's many ways to build better models, but that's the gist of what you're going to want to do as a scientist. And then you're going to come back and reevaluate those new models, maybe against the old data plus any new data that you collect. And then you're going to go the other way and build a new ANN model. And this is the process that ultimately will produce models. And the claim that I want to make here is that the best models are the best scientific hypotheses. That's the output of this process here, of this back and forth model test, model test. Those are, I'm going to call them mechanistic scientific hypotheses. And I want to sort of highlight what I mean by best is not, hey, they do computer vision well. I mean, they best match all the available empirical data, both neural and behavioral. That's what I mean by best, and I'll say much more about that later. I want to emphasize, and this is in a way some have miscast our work here, that when we do this kind of science, we are claiming that the models are the scientific hypothesis, not the data. Those are not the hypothesis. The images are not the hypothesis. The ways we evaluate the models are not hypotheses. They're just evaluation methods. And the ways we even optimize the models, they are not our lab's hypotheses about what's going on. There's some people in the field's hypothesis, but we just think of optimization as a way to get to something that's a model of the system. Okay, that's the framework you should think about in everything I tell you today. One other thing I want to emphasize is that the science here is never finished. So it's a process. It goes on. And to paraphrase George Box, no model is perfect, but some are useful. And the rest of my talk, I'm going to try to emphasize for you that some of these models, even though not perfect, are already useful, not discounting the fact that we also need to keep making the models better. So those are the two forward-going activities if you accept this philosophy of how to proceed. Okay, so because science is never finished, what you should think of is that the current best models, now I'm emphasizing current, are the current best scientific hypothesis. There will always be a better scientific hypothesis. And when I say current, I mean those that currently match the currently available brain and behavioral evaluations. And there'll always be more experiments that can be done to try to improve such systems. Okay, this is not just philosophy. This sounds like philosophy in some level here, and I hope it partly is, but it's not just philosophy because we've been deploying this. And so I'm going to come back to our problem, which I described earlier, the ventral stream, and how to proceed on this systems level problem. And I'll show you kind of a track of how the field has been actually executing that strategy and where it's going to next. So you may know this, but models of the ventral stream existed well before I started working on this. This is far back as the 1980s. Models of Fukushima and others, motivated by early work in Hubel and Wiesel and Vision, built a series of models, some of which you see here. I'm not going to go through the details of them. One of my favorite ones is the HMAX class from my colleague Tommy Poggio, shown in the middle. These were inspired by the neuroanatomy and the functional physiology of the ventral stream. And they were our initial start models of our field, if you will. And they were all essentially neural network models, and they had neural components that could be mapped into the brain. Okay, then so what you want to do, if you follow my philosophy here, is that the useful model should align with the neural data. What neural data should it align with? Well, we are especially interested in the kind of neural data I just described for you, the patterns of firing rates out of high levels of the ventral stream. Here's one of those example IT neurons that I described earlier. I showed it in green patterns. Now it's shown on the response on just a black line here, where each level of the black line is the response of the IT neuron for each of many different images. Here's 1,600 images shown here. This is not time. These are just different images. And you might think of this neuron as sort of preferring most of the chair images better than the other images, but not all of the chair images. And it likes some images of the planes you'll see. It's in fact very hard to ascertain with just your eye what actually makes this neuron go. In fact, that was one of the challenges in the field. Similarly, if you look at an earlier visual area, an area V4 that feeds IT cortex, a mid-level visual area, the neural response patterns also look complicated, even perhaps more complicated here, not even a hint of category grouping within these kind of neural responses. Complex response properties. Okay, those are some of the response properties that need to be explained. They're not the only ones that need to be explained, but the ones we were especially interested in because, again, we had linked those to some cognitive behaviors of interest that I described earlier. So the evaluation of the first generation ANN models, here's what it looked like when we first started doing that. On the y-axis is now shown the fraction of visually driven neural response that's explained. And I should say a bit more about this. What this means is a now standard approach in the field that we applied at this time, and it's not the only approach, but the one that's applied here, is to take any level of the model, let's say a model's proposed V4 level in this case, take all the artificial neurons in that level, combine them in a weighted sum to try to predict that individual recorded V4 neuron, and then test it on some held out images. So explain means predicted for new images. And then you can do that over many neurons, and this plot shows the median over some set of V4 neurons shown here. And I also want to point out that the ceiling level here is already the experimental noise limit. We can't expect the models to do better than the limits of the experimental data. That's not fair to the models. We'll also emphasize that's today's experimental limit. Again, those limits can be improved with more data. But what you see already is that the cortical area V4, the models, 0 is basically a chance guessing model, 0%, and 100% is your perfect model in this framework. And you see the models are closer to guessing than they are to perfect in cortical area V4. These are, again, the initial generation of models of the ventral stream. Another way to put this is they have poor brain alignment. And here's an example in red of the model's predicted response over those set of images compared to the actual responses, again, that I showed you earlier in black. And you see, again, that's visually what that level of alignment quantified looks like. Okay, this is not just V4. Here it is in IT. Even poorer alignment, even closer to guessing, which is not surprising. This is one level down in the system, is going to be even more nonlinear. So these models were also not so good at IT. Okay, those are the initial generation of models, but then there was this new generation of models. I'll call them second generation ANN model building. And the way those models came from is they didn't just throw out the whole modeling enterprise. Remember this back and forth that I described earlier. They took those initial start models, which were essentially deep feed-forward convolutional neural networks, like the one shown here. Again, HMAX was of that type. Of the models that I showed you were of that type. They used those kind of models. They implemented that same basic architecture. Didn't throw out what we thought we already knew, but the key ingredient that they added was this one. They took constraints that were more top-down now. They said, let's get these models to actually do something useful, like be able to recognize a person across transformations in size and position, or a car across such transformations Those are the core recognition tasks that I described earlier that we think the ventral stream carried out. And we just made that real. We said, let's try to get these models to perform on those tasks, not just align with the brain with a first principles guess of how it might work. When we say we, the people who deserve the most credit for this in my lab are Dan Yeamans, who's now at Stanford, and Ha Hung, who was a graduate student at the time. And what we did here was to use engineering tools to optimize the unknown parameters of the model, the architecture, both the microparameters, which you can think of as synaptic weights, and also the hyperparameters of the model here. And those of you who work on artificial neural networks know there's a very large base of parameters in here. But think of the engineering as just a way to optimize towards an ethological goal that we guessed at the top. This approach, I want to emphasize, is not just training on one task. I know a trend in the field right now is to say train on multiple tasks. I think that's a good trend. But I want to remind you that in the visual system, already these models were trained effectively on multiple tasks. They were not just trained to do people detection. They were trained on thousands of tasks if we trained them on, for instance, ImageNet. And that's an important, we know that that matters for producing feature sets that are general and aligned with the brain data better than if you just train on smaller numbers of tasks. Okay, here's the output of that. This is old story for those of you who follow this area. These models were far better than those first generation of models. The first generation are black. The second generation that I described for you are in red. This was actually the early members of the second generation, and models have gotten even better since this plot. This plot is from about 2014. So we saw these big leaps in the alignment with the high levels neural activity in Varia V4 and IT by taking this kind of newer modeling approach, but we had to check that alignment again as part of the scientific process. Okay, so big picture, what I'm telling you is we were asking, are these models aligned in IT? Are they aligned in V4? And I showed you some of those data from that early work on this. And again, we found some impressive alignment with some networks that were appearing around 2013 from our group and from other groups. And those from other groups actually turned out to be better aligned with the data. And we were very happy to see that and emphasize that. We also asked, is this behavior aligned better? And we and others asked that later on, and that turned out to be actually better aligned than earlier models as well, but still not yet perfect. And then others have asked in area V2, and Andreas Tolias and others, and Matthias Bethke and others have asked in area V1 how well these are aligned, and they see pretty good alignment there as well. Now, we can continue to ask alignment questions. In fact, that's actually the direction that our group is mostly taking, is to try to keep improving these alignments at all levels of the network. And I'll return to that at the end as to how we're doing that. But I want to step back for the broader audience to say, the way you should think about these second generation model building technique that I described, it's not just in vision that you see this happening. So this dialogue between natural intelligence and artificial intelligence is really kind of being amplified by this approach where natural intelligence brings to the table usually anatomy and tasks, like I described for vision. Engineering brings performance optimization tools to optimize the networks and implement the networks. And then those networks return as scientific hypotheses that can be evaluated in the way that I just described here. And you see that happening not just in vision, not just from my group. I've listed some of the key other groups that are making moves in this direction. I'm sure there are many others that I'm not listed here, and I apologize if your name's not on this slide. Auditory groups, somatosensory decision-making, and so motor planning, navigation. So there's a broad push to do this across various areas of neuroscience. This approach has really caught on. Okay, so I want to now sort of shift to sort of kind of give you kind of a state of, that's really all by way of background for those of you who've been following this area. So where are we now? So over the last 5 years, we and others have evaluated dozens of these ANN models on primarily using those linear regression methods for the neural alignment checks that I mentioned earlier, and the image by image behavioral confusion patterns is the way we've been evaluating for most of the behavioral tests and tests like core recognition that I tested you on at the beginning of the talk. And the way that sort of upshot, the big picture summary that I would give of the field and ventral visual stream models is that it's a glass half full, half empty story. And I think it's important to not, try to say, oh, it's full or it's empty and try to battle between that. The reality is it's a half full, half empty story, and that some ANNs, some of these models, not any ANN, just some particular ANNs are the demonstrated leading models of coding in the primate visual system. And what I mean by that is they are the models that best align with the neural response patterns and the behavioral response patterns at multiple levels considered simultaneously. And that's good news. That's the half full part. However, we know that no model aces all the brain and behavioral alignment tests. We and others have shown that that's not perfect in areas V1, V2, all the areas, none of them are perfect, and the behavior is also not perfect. So that's the half empty part. Okay, so where are we then? Where do we do next? So that's sort of a summary background of kind of what's happened in the ventral stream modeling and the interplay between modeling and data, just broadly at a high level for all of you. So where is the field now? Well, so many of you may know there's a lot of debate going on about these models and whether they should count as understanding. We don't have really good theories of what makes them work and what conditions they'll fail. That's an active area of research in the ANN field broadly and machine learning as well. That's not the area that we are most pushing on to try to get to those theories. We hope we can benefit from them at some point, but we don't think we necessarily need them to proceed with the progress that we hope to make as neuroscientists. So another way to look at this is to say, is the current best model, should they even count as an understanding? And again, that relates a little bit to the idea of theories or what the word understanding means. Or are they merely a simulation of the ventral stream? And I'm putting merely in quotes. And we would say these are a simulation, but they are a very useful simulation, and therefore we're calling them an understanding. So we're seeking a very powerful simulation that can be used for the goals of what an understanding should be. And I'll say more about that in a minute. We ask, what can we do? Instead of debating whether this is a sufficient understanding for other humans to transmit to each other, we ask, these models as transmitted at the moment, what can we do with them? And that's a judgment of how good of an understanding they are. And so the way you should now step back and think of this back to the philosophy is that anytime you're building models in engineering, again, some models are useful. So what does useful mean? Well, that means you can deploy them for some kind of applications. Now, and many of these systems that are related to this are actually deployed in things like computer vision and others right now. I'm going to tell you about sort of our moves to try to take some of these ventral stream models and start to make progress in little bits of applications. None of these is yet a full real world application, but they're all pointing in the direction of various types of application. What types? What types should we imagine? Well, for us, one is neural control, and another is brain-machine interfaces. Another is, and those both relate to kind of ideas of how you might improve human health. Others relate to computer vision and AI, and that's, I think, obvious for the setup of this talk. And others relate to possible kind of applications in education and visual learning. So I'm not going to tell you about our work in all these, but I'm going to give you a sort of a vignette on each of them so you get a flavor for what we're doing here and where these models are useful. So neural control. Well, this story really, I'm going to just briefly say, because this was 2 years ago, a couple of great postdocs shown here, Puya Bashvan and Kohita Kar, got together and did this test of neural population control. And I'll show you briefly what they did. This is published work again. If you're interested in the papers at the bottom, the idea goes like this. If you have a really good engineered model of a system, like the visual system, then you can pick a control goal, like you want to set the V4 population state in a particular state. And you say, I just want to turn all the neurons on or turn one of them on and all the others off. Whatever goal that you have for controlling the neural state, you can make that a goal. And instead of trying to do that directly with probes in the brain, you can actually start to ask, what are the ways that I can custom design the pixels on the image to set the internal state to that desired goal state? So you can allow the model to search through images to effectively find new ways to drive the system into states that maybe it never visited before, or just that you desire it to visit in certain ways. And so that is something we've been pursuing as neural control. We ask the models, because we have engineered models, they're implemented. You can go back and look at the pixels. You can use the compute power to find the gradients across the pixel space, to find the directions to change the pixels so that they produce that goal state at the goal level, in this case, area visual V4. And there are lots of ways to do this, but the key point is that the engineered models allow us to start to design such custom images to try to drive the neurons into particular states. And we tested this experimentally, and we demonstrated that you can actually do this quite well. And I'll just sort of give you a feel for that here. This is sort of a very simple sort of toy example of that. On the x-axis is the model predicted firing rate, and the y-axis is the measured neural firing rate of a V4, a single V4 neural site, which is a mid-level visual area. So here the idea is each dot is a visual image, and you see how well the dots line up along the line. What that means is the model is very good at predicting the neural response rate. These are predictions on new images. Now that good alignment shouldn't be surprising to you, because that's essentially the work I outlined for you earlier, that these models have made great advances in their ability to predict the neural response to arbitrary new images. But now we ask the model to sort of go beyond its sort of comfort zone and start to extrapolate to say, drive the neural firing rate of this site up beyond what you'd observed so far. And this is a sort of, you can think of it scientifically as a stronger test of the model, but from an application point of view, again, you can think of it as a control goal. And so let's drive it up here. And we asked the model to come and find us some images that would drive it up to this corner up to the right, high firing rate. And the model sort of came up with these particular images when we worked back through the pixels to activate this neuron within the neuron's receptive field. These are some of the visual images it just happened to come up with this neuron. I don't want you to stare at the images. Again, the images are not the important part. The important part is the model is the thing that came up with the images. And the images produced these firing rates measured out of this particular neural site in this experiment. The model thought they would be pretty high firing rates, as you see on the right, and they were actually, were much higher than anything we had seen before out of this neuron over the natural image space. So that's what we call success. Although I also see glass half empty, they're not on the diagonal anymore, which I'll take as kind of, that's a partial failure, which says we need a better model and we need to fill up the glass. But you can already see that we can do things better even with the half full part of the glass than we were able to do without these kind of models. So that value add is already there in the models without the full conceptual understanding, if you will, about these models, or even theories of what makes them better in certain cases. We can already begin to do these remarkable application-oriented things. Okay, take home, I just said this, partially active computable ANN models allow us to use control knobs to precisely modulate internal neural brain states. Okay, that's kind of application pointer one. Again, not full application, but in a direction. Let me go to another area, computer vision AI. Of course, this is an area that we should be impacting if these models are good. And I want to tell you another vignette there. So here the vignette starts by thinking about kind of the behavioral level as a start point. If you think about computer vision, its goal is to really make well-behaving systems. So some ventral stream-like ANN systems they're now competitive with primate behavior on computer vision benchmarks, and that's highlighted in the popular press. Of course, if you look closely, you can already see that they're not the same as primates, and in particular, they're not as robust as primates, and I'll show you an example of that in a minute for those of you who haven't seen it. But for us, what we wanted to do is just start to improve the models, see if we can slightly improve the models and get any improvements in the behavior. So can we even make a change in these early visual levels of the model where we have a lot of neuroscience knowledge to say, let's take the models as they are, but let's try to make them even more like V1 from their current start point. And when I say we, and this work is led by a postdoc, Diego Marx, who's at the conference, and Joe DiPello, a graduate student. So what they did in this case was we focused on a particular AI computer vision need. And I mentioned this a minute ago, robustness of these systems. So those of you who don't know this already, this is widely known in computer vision, but for those of you who don't know, I need to outline it. So this is sort of the flip side of the control example I just gave you. So these models, they have this kind of weakness that if you show them this image, for instance, on the left, the ANN model might say, oh, that's a terrier. And you say, great, that looks really good answer. But you can work back through the models to try to purposely confuse the model. So it's called adversarial for that reason. And you're trying to confuse it to make a misclass guess. In this case, it's converting the model to guessing that that image is a church. And the way it's doing this is by slightly modulating the pixels with that noise image that you see in the middle, added on top of the first image to make an image that to us looks almost identical, fairly imperceptible, yet causes the model to completely flip its category output label to church. So that's considered a catastrophic failure from a computer vision point of view. It's not actually clear whether humans have these deficits or not because we don't have white box. I can't make clear white box attacks on humans, white box meaning full access to all the details with inside a human or a monkey brain. But let's assume that humans we think are more robust than these kind of systems here at the moment. And so one of the cool things that Joel and Tiago noticed is that when they just started to look at those models, and remember, there's a whole suite of ANN models. And here now, looking at the models, ANN models V1 level, so just that early part of the model, the x-axis showed its match to the V1 population pattern using metrics like the ones I showed you earlier, these like linear regression metrics. That's what's shown on the x-axis. And each dot is a model. And then on the y-axis is the robustness of that model to these kinds of computer vision adversarial perturbations that I outlined on the left. So higher on the y-axis is better, less fooled by that kind of manipulation. And what you see here is there's a pretty strong correlation between just making the V1 level better than several levels down in the network. Remember, these are deep networks. At their behavioral level, the models show improved adversarial robustness to these kinds of attacks. And that was sort of quite interesting. And we said, well, let's not just kind of notice that, but let's actually act on it. So let's update the models to make them more V1-like, make their front ones even more like what we see in the brain, sort of a better silicon copy, if you will, of the front end. And what we found is that when we do that, we get boosts in robustness without additional computational costs relative to other techniques that are used to boost robustness. And when I say we, this is a team of people, some of which are shown here, including Joel and Tiago. And what we basically did was build something so-called V1 module, which is modeled after known neuroscientific knowledge of the ventral stream, again, accumulated over decades, placed that as the front end of a neural network, place the back end similar to standard back ends that we use as models of the ventral stream that I referred to earlier, optimize the entire thing except the V1 module to perform tasks like I described earlier. And so now you have a full new model of the ventral stream, which we refer to as a V1 net. It has a V1 module in front of it, plus the rest of the network. And what we showed in this particular study here is that for any backbone architecture, whether it's a ResNet 50 or CoreNet S, the different backbone architectures, each of them led to improvements in robustness of those kind of adversarial white box attacks that I described a minute ago, shown on the y-axis. And you see each blue bar is the boost you get by putting that V1 ResNet in a fair comparison against without having that V1 neuroscience motivated block in the front end. It turned out that even though we hadn't designed it for this, we hadn't measured this ahead. It turned out these models actually had more robustness to other image variations like additions of noise, weather, blur, and digital effects that, of course, humans are reasonably good at dealing with, yet current computer vision systems were still not as good as humans. And when you take this as a whole, over all measures of robustness, these models here that we were describing turned out against fair competitors to be some of the leading models of kind of robustness across these sort of set of perturbations, including adversarial attack combined with these other image corruptions. So that was an interesting value add that we see on the computer vision side that came from just making the models a little bit more brain-like. So again, I'll put that in the bottom as sort of taking models, making them better, leading to some gains and things that are of interest to applied computer vision. Okay, last vignette that I'd like to tell you is about thinking about today's models for brain-machine interfaces. So let me give you that story here briefly. So the idea is, if you could imagine using the ventral stream models to understand the perceptual effects of direct neural perturbation. So here a brain-machine interface would be, imagine that you could put inputs into a high level or mid-level of the visual system, and say if you're a blind individual or something's wrong with your retina, yet you can still induce percepts that would be useful to your daily life. That's an example of a brain-machine interface input in visual system. This work, and really we're far from that kind of technology yet, but we're telling you here about the science that might lead to that based on these models. So this work is led by a combination of people, and some of them are listed here. Hyo Lee, who was a graduate student at the time, and Martin Shrimp, another graduate student, and Paul McGrath, a master's student. And so the idea is that you take current models. We already know, as I've just described, that they're pretty good at predicting the image-elicited neural activity in high-level visual areas and the behavioral outcomes. And that latter part of behavioral outcomes is important because, remember, we're going to modulate the neurons within the models, and we're going to use that to test the changes in behavior. Here's the setup. This is basically another slide showing you what I've already told you before. You have ventral stream at the top, actual behavioral report, and then you have models at the bottom with their behavioral report. And we have pretty good alignment, as I described earlier, kind of glass half full and building those kind of systems. But the models as currently built, as they were built at the time, they didn't engage with the neural perturbation studies. And I'll say more about that in a second, of why they didn't do that. But the idea that we wanted to pursue, sorry, I'm getting stuck now. Oh, back up. Okay. The idea I wanted to pursue is predicting if and how perception can be precisely modulated by direct neural perturbation. So could we, for instance, put in direct neural signals into, say, IT cortex as shown here, and modulate the subject's perceptual report? Again, that's the BMI setup. And what we wanted to do on the modeling side was to ask this same kind of question, to apply perturbations to the IT level of a model, for instance, and that will produce predicted perceptual effects, and we should see if those align with what the subject's perceptual effects are in experiments. And again, this is not a BMI attempt. It's a sort of a strategy to see if the models could engage on what's needed for a science of BMI. So here's how we did this kind of work so far. And again, these are early days in this work. The idea is we want to make a neural perturbation, say a single site neural perturbation in IT, which is typically how most of the experimental data are at the moment. And we needed to make the models engage with this kind of question. So the models as they existed that I described earlier, even though they have ITs, those are just big bags of neurons with functions that describe pixels to neural responses, but they don't lay those neurons out in any kind of topographic 2D space or 3D space within the brain. And so what we did, and when I say we, in our lab, this was Hyo Lee, her work on doing one variant of doing this. She actually co-trained models to both lay out neurons spatially under a sort of proxy wiring cost function, which is schematically illustrated at the right, and I don't have time to take you through, and to still perform on the kind of tasks that we described earlier. So it's the same strategy for the second generation modeling as earlier, but now putting in a wiring cost proxy to cause the neurons, at least at the IT level, to force them to have a spatial layout that sort of, we think, roughly relates to wiring cost. And this is an example of the layout of an IT of one of these particular topographic models. It's a 10 by 10 millimeter grid showing the IT neurons, looking at them top down. And here this just shows the example of these particular neurons' preferences for faces versus objects, which is an important topographic marker of IT cortex. Notice there's sort of red clumps, which are called face patches in the biology literature. And one of the things Hyo showed is that those things naturally emerge out of this kind of strategy. Anyway, others are doing this kind of approach, I should say, of topographic models. I especially want to highlight Talia Konkel's work and Dan Yeaman's group work on this as well. There's other ways to build topographic models that we think can be engaged. But we went forward with this particular brand of topographic models because we had them handy in our group. And when I say we, again, what we did was to now start to model neural perturbations that had previously been used in experiments like IT cortex. And here now, Martin and Paul started to take what was known from examples of, say, if you put mucimol into IT, these are the kinds of silencing patterns you get. Or if you put microstim or optogenetic inputs into these areas, there's known spatial distributions from those kind of experiments that we can use to kind of guide the models to what to set it up to ask it what predictions it makes on behavior. And so we then applied those in the same way they were applied for each of the experiments that have already been done in these kind of direct interventional experiments within the ventral stream of the monkey. And then we compared with the actual results of those experiments. So just conceptually, you can think of like, what if we silence this big clump of face preferring neurons within this single face patch here? What would be the behavioral consequences of that kind of manipulation? And the model can now make predictions on that to break this down for you. So a big picture summary is that, okay, we have topographic models that we had locked down. We had perturbation parameters from the literature. Again, mucimol, optogenetics, microstim, electrical microstim, different ways of intervening in the system, different possible BMI related strategies. Those were all locked down. And then we took the models and asked them to make predictions for a range of neural perturbation experiments, which are shown on the left. So in the green are some example results from some sets of experiments, and on the right are the model data. I won't take you through this in detail. I don't have time to do that today, but just I want to say that all the experimental results are at least qualitatively predicted by this approach. And if you are really interested in this, I'll just sort of say the current ventral models, they may provide a reasonable platform for BMI testing already in their current state. At least they provide a platform to guide the next BMI-related experiments, and we're trying to carry some of those out in our lab now. If you're really interested in this, I'll point you to this poster. Martin Schrimp is on site. Again, he and Paul McGrath led this work, and please go to their poster number 73, and you can hear about all of that in great detail. So I'll point you there. Okay, back in the last few minutes of my talk here, I just would like to come back to sort of the big picture and sort of I've tried to emphasize for you why this way of building scientific hypotheses are useful. It already starts to engage a range of applications that I described. But for us, the sort of the engine, the meat, if you will, is that upper part of this interaction between models and data to build even better models, because the better we can make those models, the better those follow-on applications will be. So I want to return to just this piece up here, this evaluating the current best models piece. And this turns out to be quite crucial. And I've glossed over it a bit today, but I want to spend the last few minutes talking about it. First of all, this is not easy, and it's not rewarded. This is not something our field tends to reward, but it's turned out to be absolutely crucial. So with that backdrop, remember the models that we're building, this is our particular domain of ventral stream, is trying to align with ventral stream data and behaviors that we think are related to the ventral stream. And you have to keep in mind, the reason this is challenging is because there are many areas, this is a really simplified diagram of the ventral stream, and there are many possible benchmarks and data sets within each of these areas that could be imagined. So it's not like one single number is obviously what should be measured. So that all has to be maintained, assembled, and put together. And here is Martin Shrimp again, a graduate student again on site. He deserves a lion's share of the credit for getting this started, and an open source platform called BrainScore. This is not in my lab. This is bigger than my lab, but it's something that we've contributed to, and especially Martin's really led. And basically what BrainScore is a way for modelers to submit models and people with data in the ventral stream to submit data and benchmarks, and allow a rallying point and a meeting ground to sort of evaluate models on those the existing data, all the existing benchmarks, plus any new benchmarks that others submit. And what you have are a set of models. Here's 8 example models shown on this plot. And you have some benchmark summary scores shown for different visual areas here. This is, if you're interested in this kind of strategy, I will refer you to this perspective piece that we wrote, that Martin wrote about a year ago. It gives all the sort of pros and cons of doing this and all the challenges of doing it this way. So I hope you point you to that if you're interested in learning more. But a key message from that is that data alone are not enough. We need benchmarks that are basically a mapping between the data and the model. So data plus a metric gives you a benchmark, and that's what you want to evaluate on. And that is what needs to be maintained by these platforms, not just repositories of data. And that's what BrainScore does. And BrainScore continues to grow. This is what it looks like today. There are a lot more benchmarks than you saw on that early slide a minute ago. And what you see is neural alignment benchmarks, and you have behavioral alignment benchmarks, which are broadly, to me, they're all about that brain alignment benchmarks that I described at the beginning. We also have engineering benchmarks like ImageNet performance or measures of energetics that we're working on, and that's also important. So any new model that's submitted is automatically evaluated on these benchmarks and returned to the model builder. And I just want to say here on my last slide, and I know I need to close, is what does this even enable? Why are you doing this? I mean, it feels like, it's a good thing to do, but let me give you a sense of why this is useful. First of all, it incentivizes and tracks the building and dissemination of ever more accurate models of the ventral stream. So this is what allowed computer vision to start to succeed, is having challenges like ImageNet. Our field needs challenges like BrainScore ventral stream, BrainScore.human ventral stream, BrainScore.mouse v1, examples of challenges like that to make this, to drive our field to produce these kind of reproducible models that rally us together. The other thing it does, and I think this is really what's really cool, I think, is it enables discovery of if and how subcomponent improvements in models lead to improvements in other areas of alignment with the brain that those of you modeling those subcomponents wouldn't otherwise be able to see. So an example of that, I'll highlight for you here is, remember those V1 model alignments that Tiago and others were working on. Here's on the x-axis on this plot on the right is V1 benchmark composite score. And each dot is a model, and then here's a behavioral score. So this is deep, several layers into the model, and its output producing a behavior on core recognition. And you see that improvements in models in V1 actually tend to improve behavioral alignment scores. And that's really important if you're a V1 physiologist, to say, show which parts of your work end up mattering and which parts are less relevant to those higher level goals. And so gluing those things together and providing that feedback, I think, is one of the cool things that comes out of models through a platform like BrainScore. Okay, it also enables the discovery of if and how improved brain alignment actually matters in potential engineering uses. And you may know, for instance, some of these models on the overall brain score now shown on the y-axis, they were actually driven by improvements in ImageNet performance that I described earlier. But this can go the other way. So you can start to have improvements in alignment with the brain. And I mentioned the V1 predictivity score as an example earlier, for improving that adversarial robustness as a computer vision benchmark, those white box attacks that I described earlier, as just one example of that kind of two-way interplay, or this dialogue between here natural intelligence in green and artificial sort of intelligence and computer vision in blue. Okay, another key point to make here is that the BrainScore approach, and including these behavioral and engineering benchmarks, guards us as a field of neuroscience against this problem. So this is, early flight examples of trying to build planes that were exactly mimicking birds with feathers or trying to flap their wings. Didn't do so well in flight tests. And so having those kind of flight tests, if you will, sort of in the loop, including the behavioral and engineering benchmarks close at hand, helps keep us on track as a field as we work on, say, components of V1 or V2 or other parts of the system. Okay, the BrainScore community is not just Martin. He's really helped grow this to a whole bunch of people that have contributed code and data and metrics and other things. And if you are interested in joining, please join us. This is something that is, again, a community effort that is being shaped right now. And it's a great time to help us shape that into something that everyone can be a part of. So the take-home slide I'd like to give you, the main messages I've said today is computable models are the scientific hypotheses of systems neuroscience. those that make predictions that do not require humans in the loop. These models are the most useful. The most useful models are those that are the most accurate. For brain science, accuracy means aligned with brain and behavioral data, neural and behavioral data. And my claim, one philosophy claim I have, is that the best scientific hypotheses are the computable models that are most empirically aligned with the brain. I don't think that should be controversial, but it seems to be in some quarters. This process is never finished. This is what I said earlier, and there's always a better model. In practice, the current models, even though they're not yet perfect, they're already demonstrably useful in the real world. And I gave you some examples of where that's starting to lead. This doesn't mean we stop building models. It just says that it's sort of that is one will naturally feed the other. The basic science improvements in the models will lead to the application improvements of the types I've described. And a key bottleneck for our field in terms of improving the models is the lack of shared ways to comprehensively evaluate those models against what we as a field, systems and cognitive neurosciences now, think of what those models should be able to explain. In practice, we and my lab and others are doing our best to help this ecosystem in the subfield of the ventral stream and visual processing. Again, I hope you join in to help us shape this on the BrainScore platform. With that, I'll end and say thank you.